All right, today we're diving deep, microscopic level deep, into the world of the cell to solve a really fundamental mystery. Think of it like a detective case, and it all kicks off with one big question. So let's start our investigation.
So, picture this. Imagine you're trying to find one specific person in a massive, chaotic crowd, like a sold-out stadium. That's pretty much the challenge scientists face every single day. See, inside every one of your cells is this bustling city of thousands of different proteins, and each one has a very specific job to do. So how in the world do you find and point to just one protein, your single person of interest in that entire crowd?
Will you use a super clever technique called Western blotting? Ever since it was developed back in 1979, it has become one of the absolute workhorses of the molecular biology lab. You can think of it as the ultimate forensic tool for proteins. It lets scientists pick their one target right out of a lineup of thousands.
So how does this whole molecular investigation actually work? What are the steps? Well, the process is a series of really careful, deliberate moves, and each one gets us a little bit closer to identifying our protein suspect. Okay, first things first, we've got to collect the evidence. To do that, scientists have to lyse the cells. That's just a fancy word for breaking them open, to release all their proteins into a liquid sample. Simple enough. Next up, it's time for the lineup. This whole protein mixture gets loaded into a special gel, and then an electric current is run through it. Now, the smaller proteins can zip through that gel way faster than the bigger ones, so they all get separated out nice and neat according to their size. And finally, this perfectly organized lineup is carefully transferred from that fragile gel onto a much more durable membrane, basically creating a permanent record of our evidence.
Now here's where the real magic happens. This is how we find our specific target in that huge lineup. Scientists use something called antibodies, which basically act like tiny molecular bloodhounds. A primary antibody, which is engineered to recognize only our protein of interest, is washed over the membrane. It just sniffs out and latches onto its one and only target. Then a secondary antibody is added. This one is designed to bind to the first antibody, and here's the cool part. It's carrying a special tag, an enzyme that's all ready to create a signal for us. And this brings us to the final reveal. A chemical is added that reacts with the enzyme on that secondary antibody, and that reaction produces light. It's called chemiluminescence, and it makes our target protein literally glow, showing up as a distinct band right on the membrane. The detective's flashlight has found the culprit. Case closed, right? Or maybe not. Yeah, here's the plot twist. As powerful as the Western blot is, it is notoriously, infamously difficult to get right. For every one perfect-looking result, there are probably countless failed attempts, which leads to a whole lot of frustration in the lab. This quote from the lab supplier Advanced it really captures the feeling perfectly. When Westerns work, you get beautiful, clear, amazing data. But when they don't, they are, and I'm quoting here, tortuous. They can produce some of the ugliest, most confusing data you'll ever see. Just look at the difference here. On the left, that's the dream. Crisp, clean bands, super easy to interpret. But on the right, well, that's the harsh reality for a lot of experiments. You get these faint, smudgy signals that are pretty much useless. So what on earth went wrong? And more importantly, how do we fix it? Luckily, the story doesn't end with scientists throwing their hands up in frustration. Nope. Over the years, they've developed some really ingenious solutions to these problems, slowly turning this tricky art into a much more reliable science. And these breakthroughs really focused on two critical areas. First, making the physical process, especially that protein transfer step, way faster and more efficient. And second, making sure that the final evidence, those glowing bands on the membrane, was actually telling an accurate, trustworthy story. Okay, let's talk about the process first. The old school wet transfer method meant submerging the gel and membrane in a big tank of liquid for an hour, sometimes even longer. It was slow, it was messy, it was tedious. The modern solution is dry transfer. It uses these pre-made stacks with specialized gels. And the time difference is just staggering. We're talking about going from over an hour down to as little as 3 minutes. But you have to wonder, is faster actually better? Well, according to this data from Thermo Fisher Scientific, the answer is a huge, resounding yes. They put the 1 hour wet transfer up against a 6-minute dry transfer for a protein called Q80. And the result? The dry transfer gave a signal that was more than twice as strong. So this isn't just about saving time in the lab. The new method is actually way more effective. It's a total game changer. So awesome, we've solved the transfer problem, and now we have a strong, clear signal. But that leads us straight into our second challenge, data integrity. How do we know if the brightness of that band really represents how much protein is there? Or what if it's been skewed by little errors, like maybe loading a slightly different amount of sample in each lane? For decades, the go-to solution was to use something called a housekeeping protein as a reference. These are proteins that are just always there, things like actin from your muscle fibers or GAPDH, which is key for metabolism. The idea was you'd assume these are always present at the same level, so you could measure your housekeeper and then use it to normalize or kind of adjust the signal from your actual target protein. But it turns out there's a pretty major flaw in that logic.
Yeah, as a ton of research from places like Biorad has shown, these housekeeping proteins are anything but stable. Their levels can actually change depending on the very experiment you're running. Plus, they're often so abundant that their signal just gets completely maxed out, like a camera sensor getting blasted with way too much light. All of this makes them a really terrible reference point for trying to measure other proteins.
So the modern and much better solution is something called total protein normalization, or TPN. Instead of relying on one shaky housekeeper, TPN measures the signal from all the proteins in each lane with a special stain. This gives you a much more stable and accurate baseline to work with because it's based on the entire protein population, not just one potentially unreliable member. It's like taking an average of the whole crowd instead of just asking one person to speak for everyone. So, with these modern breakthroughs in hand, the faster, better dry transfer and the much more accurate total protein normalization, we can finally reach a reliable verdict in our investigation. By using a super stable baseline, like the total protein, to correct for all those little variations that are bound to happen, scientists can now be really confident that the changes they see in their protein signal reflect what's actually happening biologically, and it's not just random experimental noise. The evidence is no longer just circumstantial, it's confirmed. And really, that's the whole story of the Western Blot. It's this incredible journey from a technique that was often seen as a frustrating, kind of messy art form into a truly robust, quantitative, and reliable scientific method. It's a perfect example of that relentless scientific drive for better, more trustworthy data. And that kind of leaves us with one final thought, doesn't it? If it takes this much work, this much innovation and rigor just to accurately measure a single protein, just imagine the incredible complexity and challenge involved in mapping out the entire intricate web of interactions that makes up life itself. It's a powerful reminder that in science, getting the details right, it's everything. Thanks for tuning in.